Thank you, Alan, for your nice introduction. Ladies and gentlemen, if you ask physicians and pharmacists if they believe in the concept of pharmacogenetics, meaning that do they believe that genetics influences drug response, the vast majority of pharmacists and physicians answer yes, they believe in the concept of pharmacogenetics. However, if you ask them, did you order or recommend a pharmacogenetic test in the recent 6 months, only a minority answered yes. Only 4% of the general practitioners in the Netherlands has ordered a pharmacogenetic test in the recent 6 months, and pharmacists did a little better, 15%. So that may look strange. They believe in the concept, they have well accepted the concept of pharmacogenetics, but they are not using this in treating their patients. And they have high expectations of pharmacogenetics because they believe that the use of pharmacogenetics will prevent the wrong use of drug and using the wrong dose of a drug. They believe that it will detect the most efficacious drug and it will help you to decrease the incidence of side effects. As Mary pointed out, we will not end up with thousands of randomized clinical trials showing the effectiveness of pharmacogenetics, but there are 6-7 randomized clinical trials have been published in very nice journals, summarized on this slide. And the green colored in the third column are with a positive outcome, meaning that screening, pre-therapeutic screening was beneficial for the patient, and the red colored were neutral or even negative. So we did one of these randomized clinical studies with Marieke Koenen as the principal investigator. Almost 800 patients with inflammatory bowel disease were included in this RCT. Patients were treated with mercaptopurine or azathioprine and tested for TPMT. The patients were randomized to screening versus no screening. And when they carried a genetic variant, we reduced the dose by 50% of the normal dose, or in homozygous patients, we reduced the dose to 90% of the normal dose. The primary endpoint was leukopenia, and in this study we found a tenfold reduction in leukopenic events in the patients who carried a TPMT variant upon dose reduction. So in this study was really convincing, and TPMT testing was beneficial for these patients. However, a problem of this type of studies and this type of gene-drug pairs is the number needed to genotype, meaning how many patients do you need to test, to screen, to prevent one from having a leukopenic event. And for this study, this topic trial for TPMT testing, in this setting, we calculated the number needed to genotype, and it appeared to be 200. And the reason, of course, is that the absolute risk reduction is only modest, 7.4% versus 7.9%, and the frequency of the genetic variant is relatively low. So the number needed to genotype 200, you need to genotype 200 patients to prevent one from having a leukopenic event. So then we move from single drug gene pair testing to panel, PGX panel approach. Mike Dunnenberger nicely showed that if you have a panel, if you compose a panel of the most important pharmacogenes, and you include the genetic variants, about 50 genetic variants in these genes, almost every patient, 95% of the patients, carry at least one actionable genotype. Actionable meaning that if patients having prescribed a drug metabolized by this gene, they need a dose reduction or a switch of the therapy. So some years ago, we started the Dutch Pharmacogenetics Working Group, which is a 12-member multidisciplinary working group. We had two aims. The first aim was to develop, similar to CPIC, to develop pharmacogenetic dosing guidelines based upon the systematic review of the literature. And the second aim was to integrate these guidelines in the electronic prescription prescription systems used by physicians and by medication surveillance systems used by pharmacists in the Netherlands. Until now, 2018, we have composed 94 gene-drug pairs, and it appeared that 47 of these 94 gene-drug pairs were actionable drug-gene interactions. In the Netherlands, we have infrastructure available that, in a way that all these pharmacogenetic information is integrated in the workflow of physicians and of pharmacists. So all general practitioners, medical specialists, pharmacists in hospitals and in community pharmacies have access, are using the infrastructure and the national database which include this pharmacogenetic guidelines. So this is a screenshot of a physician prescribing nortriptyline to a CYP2D6 pore metabolizer, and then the system generates a pop-up showing that this patient has a gene-drug, actionable gene-drug interaction, and that the dose should be personalized, in fact reduced by 40% of the normal dose. The Dutch Pharmacogenetic Working Group guidelines have been endorsed by several organizations. The European Clinical Pharmacologists have endorsed the guidelines, and the European Association of Hospital Pharmacists and Irish Pharmacists Union have endorsed the Dutch guidelines. As Mary already pointed out, we did a formal comparison of the Dutch guidelines and the CPIC guidelines, and it appeared that these two sets of guidelines are highly similar in terms of allele classification, genotype to phenotype translation, and therapeutic recommendations. And we have found some discrepancies and are working on harmonizing these discrepancies. The Dutch pharmacogenetic guidelines give an interpretation based upon a systematic review of the literature how to interpret a gene-drug interaction, but it does not tell you which patient should be tested. So, similar to the CPIC guidelines, it is very helpful in interpreting the genotyping result if the genotype is known. However, in discussions with the health insurance companies in the Netherlands, we they mentioned, well, we are not going to reimburse tests as long as you have no guidelines telling which patients should be tested. And that's the reason why we started, the Dutch Pharmacogenetics Working Group started with introducing a clinical implication score. And this clinical implication score was set up and includes the clinical effect of the gene-drug interaction, so the severity of the side effects, and you can we can score 1 or 2 pluses. The level of evidence is taken into account with a high level of evidence with 3 pluses. The number needed to genotype is taken into account, and also if information is included in the EMA drug label, yes or no. That gives you results in a total score, and when the sum of the score is 6 to 10 pluses, then the Dutch Pharmacogenetics Working Group considered this gene-drug pair, this pharmacogenetic test, as an essential test. So we have come up with 3 categories: potential, beneficial, and essential, based upon the number of the number of pluses in the clinical implication score. I will give you some examples of scores. So we are in the process of scoring all our 47 actionable guidelines. And for example, we consider the capecitabine 5-fluorouracil and DPYD testing as essential, an essential test which should be performed before a patient is prescribed one of these drugs. So we started some years ago. We started with an implementation of pharmacogenetics in primary care. With 200 patients were included and preemptively genotyped. We set up a panel of genetic variants, including the most important pharmacogenes. 40 pharmacies in the vicinity of Leiden collaborated in this project, and we found more or less the same results as Dunnenberger. 90%, almost 90% of the patients carried at least one actionable genotype when using this panel. Patients were, so this was integrated in the workflow of the physicians and the pharmacists. Those recommendations were provided to the healthcare professional, and we found that over 85% of the recommendations were accepted by the pharmacist and implemented by the physician and implemented in the drug prescription. So we did a sort of extrapolation based upon national prescription data in the Netherlands to see what the impact would be of introducing such pharmacogenetic passport nationwide. And we found, based upon on prescription data, national prescription data in the Netherlands in 2016, that 1 in every 5.3% of the first prescriptions need a dose adjustment or a switch. And this was somewhat surprising to most of the pharmacists and physicians because they considered pharmacogenetic variation as a very rare event and not realizing that 1 in every 19 first prescriptions needed a dose adjustment or a switch. So this was all about the pharmacogenetic implementation in primary care and community pharmacy and by general practitioners, but also at LUMC in the inpatient care in our hospital. We have done several programs for implementation of pharmacogenetic in clinical practice. And I will give you an example on DPYD testing and the use of 5-fluorouracil and capecitabine. These are very commonly used drugs, prescribed drugs for the treatment of colorectal cancer, breast cancer, head and neck cancer. And the use of these drugs is characterized by a relatively high incidence of grade 3 or higher toxicity, severe toxicity in 15 to 30% of the patients. And this toxicity is even lethal in up to 1% of the patients. There's a very important enzyme, dihydropyrimidine dehydrogenase, DPD, encoded by the DPYD gene, and 3, 4, 5% of the patients have a deficiency of this gene and are therefore unable to metabolize these drugs. There is ample evidence from the literature showing that if you reduce the dose in patients carrying the genetic variants, the STAR2A variant in the DPYD gene, and you reduce the dose to 50% of the normal dose, that you are able to, let's say, to reduce the incidence of toxicity let's say to baseline. And with baseline I mean patients not carrying a DPYD STAR2A variant receiving the normal dose. So this is convincing. It is just this is a prospective study, but there is much more evidence available in the literature that DPYD testing therapeutically is beneficial for patients. So our chair of the oncology departments considered in 2013 DPYD testing standard of care, and in fact this is the way it often goes in a hospital, in an institution, having one medical specialist, a champion, who considered this test essential for all of his patients. So we started an implementation program testing for 4 genetic variants in DPYD. We're using a gene activity score, so based upon the variant found in the patient, we will reduce the dose by 25, 50, or 75% of the normal dose. So we started a routine pre-therapeutic DPYD testing in April 2013, and we did a retrospective analysis of the first 300 patients after 18 months. And in the end, so we started with a full adherence, 100%, but at the end, finally we found an adherence of 90 to 100%. So 90 to 100% of the patients who received capecitabine or 5-fluorouracil were pre-therapeutically tested before receiving the drug. Interestingly, we did not see any grade 3-4 toxicity in patients with an initial dose reduction. So when patient had a dose reduction and no toxicity was seen in the first or the second cycle, medical oncologist titrated the dose up to normal, and in that case toxicity was seen in most of these cases meaning that this titration to the normal dose in carriers of DPYD variants is not a very good idea. But I think this program shows that DPYD screening is feasible in clinical practice. So this local study at one institution was expanded to a national at a national scale, and we received funding by the Dutch Cancer Society and Alpe d'Huez funding organization, and we designed a prospective study with aiming at upfront DPYD genotyping nationwide with 18 collaborating centers, hospitals in the Netherlands. The results of this study is impressive. Will be published in the Lancet Oncology in a few weeks. We also started a pharmacogenetic counseling outpatient clinic at LUMC. Patients are seen by a clinical pharmacist and a clinical geneticist, and patients screened have many questions about their test results and are therefore offered genetic counseling. They have very variety of questions. For example, if a patient has received a test result being a CYP2D6 poor metabolizer, patients wonder for which drugs this is relevant, if this is relevant for food products, if children or parents should be tested, and if they should be retested in 5 or 10 years. So we found this genetic counseling of patients with a pharmacogenetic test result very useful and providing very useful information for patients. So we have recently, 2.5 years ago, we have started a large European EU program on implementation of pharmacogenetics, U-PGx Pharmacogenomics. It's a 50 million euro project funded by the European Union. It started 1st of January 2016 and has a duration of 5 years. We will implement pharmacogenetic testing using this pharmacogenetic panel approach, a pharmacogenetic passport in 7 European countries. In the randomized clinical trial design, and this study is aimed and powered to find a reduction of the incidence of severe adverse drug reactions by 30%. So many countries from the 10 European countries from the European Union are collaborating in this large project. We are very glad with and happy to have a scientific advisory board. Many of them are here. Mary and Dan Roden are members of the scientific advisory board. So this slide shows the project outline of the U-PGx project. The first year we have been working on enabling tools, making ICT tools, PGX infrastructure for genotyping. We have translated guidelines and have set up training and education programs for healthcare professionals and lay people. In fact, the randomized clinical trial includes a total of 8,100 patients, and we will assess patient benefit and cost effectiveness and process indicators of implementation. We have several research work packages, including next generation sequencing and also system pharmacology. We have introduced the so-called medication safety code card, so every patient who has been genotyped will receive this credit card-like cards, medication safety code card. And this safety code card contains a QR code. The QR code can be scanned using your mobile phone, and it brings you to the website of the Dutch Pharmacogenetic Working Group and gives an interpretation of the results, the genotyping results of this patient. We have defined a panel with 13 pharmacogenes with a total of 56 genetic variants, which are included in our U-PGx study. The acronym of the study is PREPARE study, Pre-Emptive Pharmacogenetic Testing for Preventing ADRs. 7 institutions are recruiting patients. They start either with pharmacogenetically guiding prescribing or with standard of care and switch after 18 months to the alternative arm in a way that every center serves as its own control. So patients are triggered for inclusion in this study when they are prescribed one of the drugs from the list for which we have a guideline available. Then patients are included in this study. So in that way, it is not really preemptive, because preemptive means that patients are tested without any prescription. But for logistic reasons, we have chosen that patients are triggered by the first prescription from a guideline for which we have a guideline, and then patients are recruited for this study. Patients are contacted after 2, 4, 8, and 12 weeks by research nurses, and adverse drug reactions are inventoried using an ECRF. We're also using an online survey system from our Dutch pharmacovigilance monitoring system. Patients can fill in forms, surveys about their experience with the drug and about adverse drug reactions. So if you divide the available guidelines, Dutch guidelines, into whether they prevent toxicity or are aimed at improving efficacy, it was shown that the vast majority of the gene-drug interactions relate to prevention of drug toxicity, of prevention of adverse drug reaction. And for this reason, we defined for the U-PGx study, we defined adverse drug reactions, incidence of adverse drug reaction as the primary endpoint of this study. So for every adverse drug event, severity will be scored using the CTC criteria. Causality will be assessed using the Liverpool Causality Assessment Tool, and then the drug genotype association will be assigned, ending up an incidence of adverse drug reactions, and we will compare the incidence of adverse drug reactions in the active arm as compared to the control arm. And as I said, we are aiming for a 30% reduction of clinically relevant, the more severe adverse drug reactions. So we are measuring a series of secondary endpoints, including cost effectiveness of this pharmacogenetic approach. And so where are we now with what's the current status of this study? We have reached 1st of October. We have reached the time we switched the arms. So we are a little 1 month after the switch, and until now we have included over 3,600 patients. But we're aiming for 8,000, as you know. So one, I think, very interesting development is the use of repurposing of whole exome sequencing data for collecting data for your pharmacogenetic passport. So whole exome sequencing is, of course, routinely used for the diagnosis of monogenic diseases, and at our institute we have data available of at least 2,000 individuals. And we wondered whether it would be possible to collect the data from this U-PGx pharmacogenetic passport from this whole exome sequencing data. And Maaike van der Lee will present a poster session on Friday at a 3-hour session with the poster number 638. The results of this study, we asked 230 individuals if they were interested to have their pharmacogenetic passport from their whole exome sequencing data, and 166 gave consent, and we built a bioinformatics pipeline to collect this data from their whole exome sequencing information. So that was highly successful. From the 56 genetic variants, we were able to collect 51 of them, 3 HLA. In the HLA gene, we were not able to collect these data, and the CYP2C19 star 17 allele and the tandem repeat star 22 allele in the UGT1A1 also we were not able to collect this data. But I think this shows some future developments where, well, let's say the discussion about cost effectiveness is not so relevant anymore, because this is data from whole exome sequencing data already available and can be used for collecting data for your pharmacogenetic passport. So I hope you are convinced that implementation of pharmacogenetics in clinical practice is feasible, is effective. This U-PGx project will provide quantitative and quantitative data for the new model, panel approach model, preemptive testing in personalized medicine, and we hope that the U-PGx project will provide healthcare professionals in the EU with the tools allowing them to truly embrace the concept of personalized medicine. Thank you very much.
So Mary or Henk, what is the process for adding new variants to the guidelines? So you have a process for evaluating genes. But as we get more and more sequence data, we're going to start to get a flood of variants. And what level of evidence will you accept for adding a variant into your guidelines?
Yeah.
You want a clinical trial for every new variant, right?
Yeah, so that is a very...
Accepting vitro data or what?
I acknowledge that we have to formalize what requirements there are for variants to be included. But the process for updating the guidelines is straightforward. Those are tables that are downloadable on the website, and we just update them on the website however as needed. But we have recognized that among different guidelines, there does seem to be a different threshold for actionability for some variants that have only in vitro data associated with them and no clinical data at all. So sometimes that comes down to the severity of the disease, the consequences of getting it wrong, the availability of alternatives. So when Bob Diazio thinks that there's a DPYD variant that is very probably inactivating, and he says if it was my mother, I wouldn't give her 5FU. I know, but I mean, we are going to try to write down more clearly what all the criteria are, because we would like it to be standardized, and it's probably not 100% standardized yet.
Yeah, so in fact we started very simple. So if there was a clinical study showing that this variant showed clinical effects, then it was included. But for example, in CYP2D6 in DPYD, new variants are found, and you cannot expect clinical studies showing this specific variant has a clinical effect of no. So you should rely on preclinical or clinical data, but that is not very well covered in our procedures right now.
Get together.
Sure.
Other questions? So maybe I missed this in your secondary outcomes, but of course for things like DPYD, where you're attenuating the dose to prevent the adverse reaction, the other side is we may have less efficacy.
Yeah.
Are you going to look at that as a secondary outcome, or is that...
Yeah, so for in the U-PGx study, this is not a secondary outcome, but in the DPYD study, which will be published in the next week, this was studied. So we saw in the group who had had a dose reduction, the efficacy was similar, and we also checked this with 5FU and metabolite levels, and the levels in patients having a dose reduction was similar to patients receiving the normal dose with carrying no variant. Yeah.
List of assays, are they going to be published somewhere?
Yeah, so in the Yeah, so the question is the list, is the pharmacogenetic panel, the passport, will that be, the selection of the variants, will that be published? So it was published, a former version was published in CPNT in 2017, December issue, but we have an updated list now and we will publish it on the U-PGx website.
Any other questions? Okay, thank you so much.
Thank you.